So today I want to share a little bit of my research on nanothermometry. So the talk is going to be organized in two parts. The first part is going to review a little bit the field of nanothermometry, and the second part is going to be geared to a little bit more towards nanodiamond in particular and how they do in this field. So the first thing that comes to mind when we think about nanothermometry is the motivation. And one thing that I notice is that most of the time, the interest in measuring temperature at the nanoscale is driven by applications. And this is because, for example, in biology, a lot of the processes that are happening in the cells are driven by certain gradient that can be chemical gradient or, in this case, temperature gradients. On the other side, we also have the fact that we are now getting to a point where we can make devices that are really small. Like in this particular example, I have I put here. This device is basically made of a three single sheet of atom: graphene, a transition metal, the chalcogenide material, and hexagonal boron nitride. So these are just literally three atomic sheets on top of each other. So all up is a few atomic layers. So we're getting to a point that we are making devices that are so small that knowing the certain quantities about these devices, like in this case temperature, at the nanoscale has become very important. Now, one thing that many people looking into this field are missing is that is not or overlooking, I should say, not necessarily missing, is that besides the application, what is driving really the field is the fact that we have a lot of available materials that allow us to do this temperature measurement at the nanoscale. So this is an aspect that should not be overlooked. We are interested in doing nanothermometry, not just because there are a lot of cool applications that we can do, but also because we have the capability now of measuring temperature on a scale, something that maybe 2 decades ago, it was not as easy to do. So before giving you a sort of a detailed overview of the field, I just want to clarify a few concepts that sets the framework for this talk. So the first thing is the classes of nanothermometers. So there are different type of nanothermometers: primary, secondary. The signal that you monitor to measure temperature can be of different nature. There are temperature sensors that are based on actual physical sensors that you need to locate next to the object that you want to measure the temperature of. Or there are sensorless sensors, like temperature sensors, like, for example, a pyrometer, right? So for today, I'm going to focus on nanothermometers that are secondary, which means that they need to be calibrated. They are not based on some, for example, thermodynamic equation from which we can extract the temperature. These are thermometers that we need to calibrate against a certain range of known temperature before they can be used. Of all the type of signal that one can use to measure temperature, these particular nanoparticles that I'm going to review today are mostly going to be using an optical signal. So we're going to monitor the temperature based on some fluorescent property of these materials. And of course, there are sensor that requires you to have a sensor located wherever you want to measure the temperature. Now, when we try to benchmark these nanothermometers, meaning when we try to compare them with one another, there are a few characteristics that we usually consider. So the first one is the sensitivity. Now, sensitivity is defined basically as the derivative of a certain observable as a function of temperature divided by that observable itself. Now, the reason we use this particular figure of merit is that we can compare, like I did this in this graph, a bunch of different techniques that might use different property, different observable, and we can put them all in the same graph. And in this graph, basically what you need to look at is the temperature range. So the longer a line is across the temperature range, of course, means the more this temperature is versatile in terms of temperature that you can measure. You can go from a very low temperature to high temperature, or you're only limited on a certain very small temperature range. And of course, on the y-axis, which is the important figure of merit here, the higher you are on the graph, the higher is the sensitivity of your nanothermometer. And as you can see, these dependencies are not necessarily linear, right? So it means that there are certain ranges, for example, for this one here, where at a very low temperature, you have a much higher sensitivity than you have at a higher temperature. The other thing that is important to talk about is resolution. Now, sensitivity and resolution are often used interchangeably, but they're actually a different concept. Sensitivity is an absolute quantity that goes beyond how accurate, how precise your measurement system is. Resolution instead depends on the signal to noise ratio and also on the temporal bandwidth. So resolution is a relative measurement, if you like. You can increase the resolution of your measurement by, for example, integrating for longer, by making your signal to noise ratio better. So this is something that I want to clarify, because it is very important to distinguish between sensitivity, which basically gives you your absolute abstract ability to measure something, and resolution, which is somewhat limited by what is your instrument ability to detect a signal, for example. Another things that comes to mind when we talk about resolution is spatial resolution. So we're talking about nanothermometers, so we're interested in measuring temperature at the nanoscale. So how small is this nanoscale? Well, even though some of these nanoparticles can be as small as just 5–10 nanometers, the truth of the matter is that the resolution of this technique for temperature sensing is a couple of 100 nanometers, because at the end of the day, we're always limited by the optical system that we're using to measure temperature. And as I said, almost all these thermometers are optical in nature. Now, you can argue that if you were able to come up with some sort of technique that allows you to locate your particle before you measure the temperature, for example, with the TEM or SEM, then you could achieve higher spatial resolution, but that's usually not done, because in reality, you tend to have an optical system, some sort of microscope to measure this temperature. So it's not always possible to have a second type of measurement to increase your spatial resolution. And one last thing that comes to mind when we talk about resolution is, of course, temporal resolution, that is related again to the signal to noise ratio and your ability of your system to measure a certain signal. Temporal resolution is very important because, of course, we would like, and I'm going to make a comment towards the end of the talk, to have this measurement to be very fast, ideally in the microsecond range, which means that it could really start probing, for example, some cellular processes as they occur. All right, and the last thing that we need to consider, which is very important, I'm going to mention this a couple of times across this talk, is what is called thermal equivalent noise. So because most of these sensitive techniques are based on measuring, for example, the fluorescence of this sensor, we need to be able to distinguish when a change in fluorescence is actually due to a change in temperature, and when there are other factors, for example, if there is scattering or if there is absorption of light. So a drop in fluorescence might be not due to, for example, a drop in temperature, but it could be due to the fact that this particular medium is absorbent or scattering at that particular wavelength. So this is something that we need to absolutely take into account, because it's one of the biggest sources of uncertainty on our ability to determine the temperature at the nanoscale of a certain object. All right, so I'm going to quickly review what's the state of the art of some of the different nanothermometer currently existing. The first, and probably historically the very first type of nanothermometer that we had, that was developed, is based on organic dye. These are basically organic molecules that are fluorescent. And the dependence on the fluorescence with temperature is basically explained by this Jablonski graph, where you can excite your dye from the ground state to an excited state that can be either a singlet or transferred by internal conversion to a triplet state that is usually longer lived, because to transit back to the ground state needs to have a change in spin, which makes the transition optically forbidden. And since you have a longer lifetime, this can be affected by external factors. And this external factor can be temperature, but also, for example, pressure. If you have a high concentration of oxygen due to a higher pressure, this oxygen can interact with your molecule and quench its fluorescence. So based on this, we can have graph of this type where the fluorescence of of a solution in which you have a bunch of these molecules can depend on the pressure, like I shown here in blue, or the temperature. And by keeping, for example, the pressure constant, then your only variable becomes temperature, and vice versa. If you keep the temperature constant, you can then monitor the pressure. And this was done in this nice little cartoon that I have here. This is a model airplane, and they measure the distribution of pressure of this model airplane inside the wind tunnel. And you can do the same for temperature. Here in this case, they use these dyes and their dependence of this rhodamine, the fluorescence of rhodamine as a function of temperature in a certain point of a microfluidic channel, highlighting how in certain points the running of the fluid was heating up the microfluidic channel in points, as you can imagine, where there are sharp turns. This is just a type. There are a lot of other types of organic dye-based molecules that can be used for thermometry. For example, a molecule that can exist in monomer and hexamer state. The ratio to which depends on temperature, and they have different emission wavelengths. So you can basically monitor the ratio between the amount of monomer with respect to the amount of hexamers, and you can extract in that sense the temperature. And again, it's very important that I note that all these temperature sensor require calibration. So you have to run them in an environment where you change the temperature in a manner that you know, and then you can calibrate the sensor knowing, okay, I have this amount of fluorescence at this temperature occur. And here is another example where what is used is the polarization anisotropy of these dyes. So basically, how oriented is the polarization of the light emitted. And as you can imagine, the higher the temperature, the higher the Brownian motion of this molecule, so the less oriented the emission is going to be. So organic dyes are very well established for nanothermometry, but they have a couple of drawbacks, including, for example, the problem of bleaching, the problem of self-absorption, and the fact that because they are somewhat delicate, they operate on a rather small range of temperature, generally around room temperature. But on the plus side, they are very well established in biological studies because they're usually biocompatible, and there are a large variety of them we can choose from. So in that sense, they are quite advantageous. A second class of nanothermometers is that of quantum dots, which is probably one of the most established. And the reason is that because we can really well tune their emission wavelengths. So now we have these nanothermometers can really suits our bill in terms of if I have a specific requirement. For example, I have a specific biological environment that I know as a transparent window in a certain range. I can choose the quantum dot that I want in that particular temperature, the particular wavelength range. So in the case of quantum dots, the temperature dependence manifests in several ways. For example, in line shift or changing fluorescence, changing full width at half maximum, and the main there are a bunch of mechanisms that occur at the same time as the temperature change, including, for example, the change of the lattice parameter or the electron phonon coupling. And we can, again, these are secondary nanothermometers, so we can use a calibration curve. And like in this case, we can have that as the temperature changes, we have both a change in fluorescence, a change in full width at Al maximum, and a change in the central wavelength. And so we can monitor temperature by monitor any of these quantities, or even all of them at the same time. And now I want to mention this particular study because one of the things that it's very important when one does nanothermometry is, if possible, you would like to have a measurement that is ratiometric. What that means is that you have some way of referencing your measuring to itself. And this is because, as I mentioned earlier, sometimes you're performing this measurement in an environment that might have a high level of scattering or absorption. And so if you just monitor the intensity of your emission, a drop in fluorescence might not necessarily mean an increase in temperature. It might mean that simply your light coming from your sensor has been absorbed or scattered. And so if you have a way of referencing your measurement to something else, then you are immune to scattering and absorption. And this is what was done here with these quantum dots were doped with some manganese, and there was the fluorescence of from the quantum dot, from this ion, was opposite in direction. So the fluorescence of the quantum dot is going down as the temperature goes up, and vice versa for the ions. And so you can reference these two emission to one another, and so you now are immune to thermal equivalent noise, which in this case would be due to absorption or scattering. Now, as I say, quantum dots are some of the best established nanothermometers, mainly due to the availability of wavelengths that we can choose. They suffer from a few problems like self-absorption, because some of the wavelengths are overlapping with one another. The absorption and the emission are overlapping with one another. And being very small in size, they are also very sensitive to the chemical environment around. So if you have a quantum dot that is exposed to certain chemicals that quench the fluorescence, now you have another source of thermal equivalent noise, because that quenching is not due to a change in temperature, but is due to the change in the chemical environment. But of course, there are techniques that one can have, like surface functionalization, to reduce these effects. And of course, if you have self-absorption, you also dependent on concentration. If you have a high concentration of these quantum dots to determine the temperature in a certain area, that means that the more quantum dots you have, the more you are susceptible to this self-absorption problem. All right. A third category of nanothermometers is that of upconversion nanoparticles. These are based on basically multi-photon absorption and emission at a shorter wavelength than the one they get excited by. And the reason that they work as a nanothermometer is that at the same time, this upconversion nanoparticle have several lines, several emission lines, and some line, like in the case of the spectrum on the right, they're almost independent of temperature. But some lines are very highly dependent on temperature, and this is because some of these levels are thermally coupled to one another. So there is basically a Boltzmann distribution, and that means that as the temperature changes, some of these levels start talking to one another, and the fluorescence become less efficient, and that's the case for this graph on the left. And in other cases, this level are well protected and isolated, and they're basically temperature independent. And so that's very good, because then we have, as I said earlier, a way of having a ratiometric measurement. We can take the ratio in intensity between one of these temperature-dependent peak and one that is independent of temperature, and then we can have nicely plot this ratio plotted over the temperature and get a nice response. So upconversion nanoparticle, like quantum dots, are well established as a nanothermometer. They have a few problems. One of them is the fact that they get excited in the infrared, And if you're doing measurement in biology, you always have to be very careful, because if you change wavelength, for example, from 980 nanometer to 800 nanometer, the absorption coefficient of water changes by 20 times, which means that you really have to be careful of heating up your medium with the laser that you're using to just simply probe your sensor. So you don't want to, as you probe your sensor, heating up your environment, because then your measurement of temperature is completely off, because it's a temperature that you are causing with your measurement. But probably the biggest drawback of upconversion nanoparticle that has come up recently as one of the aspects that might limit their use as a nanothermometer is the dependence on the laser intensity of their signal. So because it's a multi-photon absorption process, the absorption cross-section of this process is highly dependent on the laser power. And so if you have a fluctuation in the environment that are more or less sensitive to a certain wavelength, your sensor gets excited more or less, and so you're going to have a change in fluorescence. So the local environment highly determine the sensitivity of this nanothermometers based on upconversion nanoparticles. But overall, upconversion nanoparticles are widely used as nanothermometers. Okay, now that I've given this review of all the other of a selection, let's say, of other nanothermometers, I'm gonna switch gear a little bit, and I'm gonna talk a little bit about nanodiamonds, and not just what has been done, but what I've worked on myself in this field. So how do nanodiamonds fit in this big field? Well, as most of you are probably well aware, nanodiamonds are host of color centers. Here I have represented the nitrogen vacancy center, the silicon vacancy center, and the germanium vacancy center. And these emitters are fluorescing. So we can use the fluorescence of this emitter for a bunch of applications that goes from as simple as doing imaging, or as I'm going to show you in a few minutes, to measure temperature. So using nanodiamonds for measuring temperature goes back about a decade. So if you compare it to other nanothermometers, it is relatively recent. The first proposal was in 2010. There were two proposals that happened at the same time. The first one that is represented here was based on the fact that as you change temperature, you observe a decrease in temperature for the fluorescence of the nanodiamond. And it's due to the fact that if you look at the fluorescence of MV center, you know that as you excite them with green light, you polarize the system in the ground zero state by this selective decay. And the interesting thing now is that if you increase the temperature, you are messing up, if you like, in a very simplistic manner, this polarization mechanism, and so some of the fluorescence now gets quenched, because you are populating less and less the zero state that you have when you are at a low temperature. And so this is reflected by this drop in fluorescence. This was a very simple, if you like, one-to-one observation that led to proposing using nanodiamond first as actual nanosensor. Almost at the same time, people looking at this MV center scheme also thought that it was possible to measure the temperature as a variation of the zero field splitting. Okay, so I'm just going to quickly review what this ODMR measurement is for those of you who are not familiar with it, although I think that not many of you are not familiar with it. So as we excite the MV center, this transition has spin conserving. So if you are populating the zero state, you're cycling between these two zero excited and ground state. While if you are in the plus and minus state, you have two chances. Either you cycle optically between these two states, Or you can also access this metastable state that is dark, it scatters less photon. So if you are pumping a microwave field resonant with this ground transition at the right frequency, you should observe a drop in fluorescence, as shown here. So when I hit this 2.87 gigahertz transition, which is the distance in energy between these two levels, as you can see, there is a drop in fluorescence. And as you can see, as I change the temperature, this resonant frequency changes a little bit. And the reason is that if you treat temperature with perturbation theory in the Hamiltonian, you have a term, and this term basically tells you how this plus and minus one level change as the temperature change in energy. So a change in temperature produces a change in energy for this level that you can monitor on this signal here. And this proposal in 2010, a few years later, was then used to do some very breakthrough measurements. I'm just going to summarize this one here. So a couple of nitrogen vacancy centers were located inside a living cell next to a gold nanoparticle, which is represented by this X over here. Then laser light was used to heat up the gold nanoparticle and therefore the local environment. And then the temperature of the environment was monitored using this method, this ODMR shift. And of course, this nanodynamo was closer to the particle than this one, and so the temperature was monitored for these two, and they could basically figure out what the temperature was in these two different locations of the cell. And to date, this is still the highest resolution method that we have for measuring temperature at the nanoscale when we're talking about nanodiamond. After that, in the few years that came after, from 2015 and 2018, a whole plethora of study came out trying to use in nanodiamond for nanothermometry, and not just using nitrogen vacancy center, but all type of color centers in diamond. And these were pitched with the idea that, unlike the one that I just described for the NV center that needs a microwave excitation to excite the ground transition at 2.8 gigahertz, all these techniques that have been proposed and realized are all optical. What it means is that you probe your sensor with the laser, and you just monitor the fluorescence, and you extract the temperature just by looking at the fluorescence signal. So that's the advantage that you're not using microwave, which if you're for example, doing biological measurement, which is the field where nanothermometry is the most active and the most sort of developed, is very important, because you don't want to microwave your cells while you're trying to measure the temperature inside them, right? All right, so in the next few minutes, I have to convince you why, since there are so many available techniques to measure temperature using diamond, I have to come up with a different one. And so hopefully by the end of the next few minutes, you realize that the idea that I had is somewhat meaningful. So the idea that I had is actually quite simple. And in fact, so simple that I was quite surprised that nobody has done it before. So usually when we are exciting our color center, we are just represented here the emission spectra of the MV as a reference, and in black we have the absorption spectra of the MV, which I have simply done as a mirror image of the emission spectrum on the zero photon line. So what we usually do, we excite with the high energy photon, in this case, for example, in the green, and then we observe emission at longer wavelengths. That's all well and good. And the lost energy is usually lost in some sort of vibration or phonon in the surrounding host or surrounding environment. So the idea that I had was, well, can we do this process in reverse? Can we excite at a very long wavelength, way past the zero photon line, and somehow hope that we can absorb phonon vibration from the environment, because that energy is available. So these phonons are zipping around our host, our diamond. So hopefully it is possible that the conditions are met by which on top of my excitation, I can absorb one of these phonons, and then I should be able still to see emission. So if this works, and it might not, this would be very good because, of course, this process is dependent on the phonon density of state multiplied by the transition amplitude. And to make this in simple terms, these phonons follow a Boltzmann statistic, a Boltzmann distribution, which you well know strongly depend on temperature in an exponential manner, which means that I should be able to have a sensor that depends exponentially on temperature. Now, of course, this is at the moment is all an idea, because one might argue that this process that needs to pick up a phonon from the lattice is very inefficient. So does it work? So here is, we tested this with 3 color centers, the germanium vacancy center, the silicon vacancy center, and the envy center. And in this graph, what you see is the emission spectrum of these 3 color centers. And in green and red, you see the traditional, what I would call Stokes excitation. And in red, I represented what I call this anti-Stokes excitation at longer wavelength past the zero phonon line of the emitter. And so the question is, do we see emission when we excite past the zero phonon line? So the answer is yes. So these 3 graphs basically show this green band represent a band pass filter that we use to filter out the emission from these color centers, and the blue and the yellow represent excitation under traditional Stokes excitation, which of course we know produce fluorescence, and the one that instead relies on anti-Stokes excitation. And as you can see, in both cases, we can see a line. Now, don't be fooled by the fact that this line have the same intensity. These I have normalized them for representation purposes. In reality, there is a factor 10 to 100. So this anti-Stokes excitation process, where we excite the color centers with longer wavelength, is between 10 and 100 times less efficient than a regular excitation, as you expect, because as I mentioned earlier, we need to be lucky enough that on top of having our light exciting the center, we need to pick up phonon from the crystalline lattice. However, as inefficient as it might seem, this thing is efficient enough that we can do anti-Stokes excitation of single color center. So here what you can see is a bulk diamond that has been irradiated to have germanium vacancy centers in it. And the one that I circled here happens to be a single photon source, so a single GeV. And as you can see, this is a spectrum of excitation with an anti-Stokes excitation, so a wavelength above 602 nanometer, which is the wavelength that the germanium vacancy emits at. And as you can see a peak. Now, I should warn you that to get this signal to noise ratio between the signal and the background, this measurement took 12 minutes, which admittedly is very long and is not good to have such a long measurement. But this is just to prove the concept, to prove that in theory you can apply this anti-Stokes excitation to even a single center and you will still see a signal. In reality, when you do a thermometer measurement, what you want is to pack your nanodiamond with as many color centers as possible, right? Because you want to maximize the fluorescence. And they're all going to depend equally on your Stokes and anti-Stokes excitation. So in reality, this long measurement only applies to a single color center. If you have a nanodiamond pack of GeV, you can just acquire for a couple of seconds and you're going to get a nice clear signal, okay? I just want to point that out. All right, so here is a little bit of the more details about the measurement. As you can see, this is just a nice representation where you can see that the spectrum and the anti-Stokes excitation, as the temperature decreases, the intensity decreases. And of course it does. As the temperature decreases, you have less phonon available in the diamond, and so you're going to have less fluorescence, because you're not picking them up, exciting your center. Remember that to excite your center, because you are exciting with very low photon energy, you need to acquire phonon from the lattice. And if phonons are not available, well, tough luck. You're not going to excite it. What I like about this measurement is that if you plot it in a logarithmic scale axis, you can clearly see this Arrhenius type behavior, where it clearly shows the exponential dependence on temperature. Now, in this graph, there are no error bars, because the error bars are basically as big as the data point. And what I also like about this measurement is that when you have a Arrhenius type graph, most of the time people do fitting both the amplitude and the activation energy. But what is nice about this system is that we only have one factor that we need to calibrate, which is this constant A, because the activation energy is simply the difference in energy between the zero phonon line and the wavelength that we're using in our anti-Stokes laser to excite the nanodiamond. So we only have one parameter that we need to fit. And as you know, when you do fitting, if you minimize the number of parameters, means that your model is much closer to what is actually happening. Now, in this graph here, I have plotted basically the same thing, but what I did, I took the intensity of this anti-Stokes excitation compared to the Stokes excitation. So what I've done here is that I monitor the fluorescence of the color center, both under anti-Stokes excitation, and then I normalize that with the fluorescence of the center under Stokes excitation. And why do I do that? For the same reason I mentioned earlier, that we want this measurement to be ratiometric. Because let's say that we have this nanodiamond inside a cell that is absorbing really, really hard at a certain wavelength. Well, this absorption at that wavelength will occur both when we excite with the Stokes and when we excite with the anti-Stokes. And so by taking the ratio between the two, we can simply get rid of this absorption or scattering effect. We have, if you like, an absolute measurement. And again, in this graph, as you can see, it follows really nicely an exponential behavior, and the error bars again are the same size as the data point. So we have a nice one-to-one correlation between temperature and fluorescence, which follow an exponential behavior, which we know will give us a very high sensitivity. And this is summarized in this graph, which I'm going to guide you through. And so here I report a few of the available techniques to measure temperature at the nanoscale, and I have benchmarked them one against each other using this relative sensitivity parameter, which again is basically how a certain observable, for example, fluorescence, changes as a function of temperature, normalized to the observable itself, such that we can compare them all together. So if you look at this, graph, a lot of nanodiamond-based nano-thermometry techniques are pretty low on the graph. They have a nice range, so they go between 150 and 400 Kelvin, which is expected. In fact, I mean, one of the main advantages of nanodiamonds is the fact that it is stable. They are stable up to a few hundred degrees, Celsius. So they are very stable in that respect. We can use them for a bunch of applications. But as you can see, these alternative techniques are pretty low in terms of sensitivity. When it comes down to it, you can resolve just maybe 1 or 0.1 degree per change in a certain observable. The one that I just talked about is this one in blue, represented by this eye, and as you can see, is comparable or slightly better than Raman technique, which we all know are some of the most sensitive technique existing for measuring temperature. Of course, one of the drawbacks of Raman technique is that you require quite a bit of material to get a decent signal. We're talking about hundreds of nanometers, maybe half a micron even, while this technique that I just proposed works with nanodiamond that can be as small as 5 or 10 nanometers, right? So if you like, the technique has the advantage of being comparable with Raman techniques, but having a much higher spatial resolution. Now, as I mentioned earlier, we should distinguish between sensitivity and resolution. I'm not going to go into too much detail here. All I want to say is that the resolution of this technique is comparable to the resolution of other system of this type, which kind of makes sense because it's basically limited by our ability in our confocal setup to determine a single to noise ratio, which is determined by the efficiency of the setups, and this has sort of an intrinsic limitation itself, and they're all comparable. All right, so this brings me to my conclusion. And my conclusion is sort of divided into two parts. So the first part is about this technique that I just discussed, which I hope shows you that we were able to develop a very sensitive nanothermometry technique comparable to Raman in terms of sensitivity, but with a much higher spatial resolution. And this technique is ratiometric, so is independent of losses like absorption or scattering from the environment. And what I like about it is that, like any other diamond-based technique, diamond has a lot of very good characteristics when it comes to doing biosensing, including the non-toxicity, the bio-non-toxicity, the high stability, the fact that you can functionalize them, and so forth. And the second half of the conclusion has more to do with the field in general about nanothermometry. So one thing that you should be concerned about is this concept of thermal equivalent noise. So the fact that when we're monitoring fluorescence, change in line width, and so forth, we need to be aware how much of the change is due to actual temperature and how much of the change is due to other factors, including, for example, the occurring of chemical reaction nearby that might quench your fluorescence, right? And this is very important. And the reason I'm stressing it so much is because I'm guilty myself of this. But when we present our result, we always say that, oh, this technique is amazing because it does this, this, and that. But the reality of the matter is that when it comes to nanothermometry, thermal equivalent noise is still something that we haven't mastered. There are we can spend an entire career trying to figure out how, in a specific case, we can remove thermal equivalent noise from our measurement, and all this work is useless when you change your sample. If you go from cell A to cell B, there might be a different amount of absorption, scattering, sensitivity to wavelength, and all the time that change will determine a different response for your nanothermometer. So that's why I wanted to stress it, because this is probably the biggest limiting factor to the ability of going tomorrow in the lab and using one of these techniques very simply to measure temperature. It's not that easy. It requires a lot of calibration and a lot of understanding and knowledge of the system that you're trying to measure the temperature. And the last thing that I want to say is about where are we going from here onwards with this field. Well, one thing that it would be nice is to move away from confocal microscopy and go to wide field type of measurement, which of course are better because they will give you a snapshot real time for the temperature of an entire cell rather than doing a raster scan. But the drawback of that is that the signal usually of this nanothermometer is quite low. So this acquisition, this wide field acquisition, might be either not very well resolved or quite slow. Which again brings me back to the idea of having one other thing that we should be focusing on is making this acquisition faster, because ideally we would like to go to the microsecond where we could then start thinking of monitoring actual processes as they occur rather than steady state equilibrium situation. Just as a quick mention, it would be nice if we were able to measure this temperature using detectors that are relatively cheap, and not just detector, but also excitation sources. Or if we are stuck with using high-end photodetectors or high-end lasers to excite and measure these signals, it'd be nice if we can combine them with super resolution technique, such that now our resolution is not anymore a couple of 100 nanometers, but it's as good as a few tenths of nanometers. Now, before I let you go, I should mention something else that I'm currently working on. This Damon is one of my children, if you like, but I'm also interested in two-dimensional material, and in particular hexagonal boron nitride, which is basically made of these single sheets of nitrogen and boron atoms stacked together and kept together by van der Waals forces, while between atoms in a plane there are covalent and ionic bonds. Now, like in diamond, these materials contain defects that emit light. And like in diamond, we can do measurements that, for example, help us measuring temperature. So in this case, as you can see, the fluorescence profile of this emitter changes dramatically as the temperature changes. And like in many other color centers in diamond, we can monitor, for example, the full width out maximum of the line width or the position of the line width. And recently we used this to measure what was the temperature of this electronic circuit. It's quite hard in this picture to see, but there are a few bands here in this electronic circuit, and there are different thicknesses, which means the Joule effect is going to be different for the different area. And we position, by using a transfer method, small flakes of HBN onto these positions, and we were able to monitor in 3 different positions the temperature. Now, the other thing that I've been working on, which is not published yet, so this is basically fresh off the labs. There is a color center in HBN, which just recently reported. It's called the VB minus. Is basically a boron missing atom in the HBN lattice. And like the MV center in diamond, this center shows ODMR. Okay, so the next idea was, can we use that to measure temperature as was proposed by Acosta in 2010? The answer is yes. So as the temperature changes, the zero field splitting, so the difference in energy between the ground zero state and the plus minus ground state, changes. And the beauty about this system is that in diamond, that changes at this rate, 64 kilohertz per Kelvin, while in HBN is about a factor 100 greater, which means that we have a much higher sensitivity. So this is, as I said, is still unpublished, is still work in progress, but of course, having more material for measuring temperature and not just temperature is of course always welcome, because one material might adapt to an application better than another one. With that, I this time conclude for real. Thank you for your attention, and of course I'll take questions if there are any.